We move on to the next topic of enzymes, which is assay of enzymes. Measurement of enzyme level in biological fluids such as plasma is often required for diagnostic purposes. However, the usual analytical method cannot be used to measure enzyme concentration because of very low concentration of enzymes and the need to isolate an enzyme from mixture of a large number of other proteins. Therefore, enzyme concentrations are measured indirectly. The velocity of reaction catalyzed by the given enzyme is measured in such conditions. The rate of reaction is proportional to the enzyme concentration. During the reaction, the temperature is kept constant by carrying out the reaction in a fixed temperature water bath or incubator. The optimum pH is maintained with the help of a buffer. The substrate concentration is kept very high. Under such condition, the rate of reaction will be proportional to the enzyme concentration. The rate of reaction can be measured by determining the rate of disappearance of substrate or rate of appearance of product. In the endpoint method, the reaction is carried out for a fixed period and the initial and the final concentration of either the substrate or product are measured. In kinetic methods, the concentration of substrate or product is measured at regular intervals for a brief period. The result in either case is expressed in arbitrary units of enzyme activity rather than enzyme concentration. So enzymes have been acting as laboratory tools also. Many enzymes are used as tools in diagnostic and research laboratories. Glucose oxidase and peroxidases known as GOD and POD are routinely used for measurement of glucose concentrations. Hexokinases and glucose 6-phosphatase dehydrogenase are used in another method for measurement of glucose concentration. Further, cholesterol esterase, cholesterol oxidase, and peroxidases are used to measure cholesterol concentrations. Lipase, glycerol kinase, glycerol phosphate oxidase, and peroxidases are used for measuring triglyceride concentrations. Urease is used to measure urea concentration. Uricase is used for measuring uric acid concentration. Peroxidases and alkaline phosphatases are used to label antibodies in ELISA. A number of enzymes are used in recombinant DNA technology, like restriction endonucleases, DNA ligases, terminal transferases, S1 nucleases, reverse transcriptase, and Taq polymerase. Certain enzymes are used as drugs also. Some human, animal, and plant and microbial enzymes are used as drugs. Streptokinase, urokinase, and tissue plasminogen activator are used as thrombolytic drugs to clear blockage of blood vessels in MI. Some digestive enzymes such as diastases, pepin, pepsin, chymotrypsin, they are used to aid digestion. However, their use is limited. Asparaginase is used for chemotherapy of leukemia. Leukemic cells are deficient in asparagine synthetase and depend upon pre-performed asparagine. Asparaginase converts asparagine into aspartate and deprives the leukemic cell of this essential nutrients. However, asparaginase is used as a last resort drug only, as remissions produced by it are not long lasting. So to summarize, we can say certain enzymes are used as laboratory tools, they are used in assays, they are used as drugs, and obviously they are used as different markers for different disorders like liver markers, cardiac marker, etc. To summarize enzymes, we can say that enzymes are of great importance. Their main function is to catalyze a reaction, catalyze a biological reaction, and only their presence fulfills a metabolic process to occur. These enzymes, when blocked or inhibited, can act as certain drugs, and they can be used as diagnostic tools also. All the enzymes are proteinous in nature except for ribozymes. So we can say that proteins, which an earlier statement is that proteins are the building block of life, is true because not only they form the cellular components and parts, but also help with the physiological function through enzymes. Enzymes consist of two parts basically, a proteinous or a prosthetic group, and apart from that, they contain a non-proteinous part which can either be a coenzyme or a cofactor. If the non-proteinous part is inorganic, it is known as coenzyme. That can be an integral part or maybe it is attached to the letter. Also, if there are certain metals which act as cofactor, which activates an enzyme or enhance the function of an enzyme. Enzymes can be of further importance because we know the processes and we know the factors which affect their activity. By controlling these factors, we can regulate the rate of velocity of rate of reaction, or basically we can regulate the reaction that is occurring through altering the enzyme in any ways. There are two theories for enzyme substrate interaction. First being lock and key model of Fisher, which is a rigid model, and the next one is Koshland's conformational change model. Enzymes are regulated by allosteric inhibitor also. They have they can either act as allosteric activator or allosteric inhibitor. When it increases the rate of reaction, then it is known as allosteric activator. And certain enzymes, they get inhibited by joining of an allosteric molecule. They are known as allosteric inhibitors for those enzymes. There are certain factors which affect the rate of enzyme catalyzed reaction, those being enzyme concentration, substrate concentration, coenzyme concentration, temperature, and pH. So with this, we complete our lectures on enzyme. Thank you.